Welcome to the Virulent Vortex, a podcast in which we discuss infectious diseases across scales, from pathogens and molecules to populations, pandemics, and everything in between. My name is Jaap de Rode, and I'm your host for this show. I am also a professor of biology here at Emory University. And today with me is Piantadosi, an assistant professor of pathology and laboratory medicine here at Emory University. And welcome to the show.
Thank you so much for having me. Very excited about this.
So, and I noticed you have really nice earrings in today.
Yeah.
They are ticks, so we're going to talk about ticks in a little bit, but I think we're going to talk a little bit about viruses and viral surveillance first, because that's something that's really close to your heart. So tell us a little bit about what that means and what you do.
Yeah, absolutely. So as you mentioned, I study viruses. I study how they evolve. I study how they emerge. And I'm really interested in sort of figuring out what viruses are out there that we might not know about, or what viruses might we know about, but sort of don't know the full scope of what they do.
Oh, okay. So obviously a lot of us are aware of viruses. You know, a lot of us get flu almost every year. When we get a cold, it's a virus. Of course, the whole world knows about SARS-CoV-2. So these are all viruses. I guess it's fair to say we didn't really know some of them before they became a problem. And so is that kind of what you're trying to find out? What kind of viruses are out there that could cause trouble in the future?
Yeah, exactly. And I'm glad you brought up SARS-CoV-2 because it's a really good example of a virus that we knew nothing about, 5 years ago. It was out there, presumably circulating in bats. We just kind of hadn't picked it up or hadn't monitored it.
Yeah.
And as you said, we didn't know anything about it until it became a problem. And it was actually a really kind of amazing discovery. There were doctors in China who recognized there were patients with pneumonia, and it all seemed a little bit unusual. Yeah. And they did some really hard work to figure out this was actually caused by a new virus that we hadn't known anything about.
Right. And so they had some ideas of, okay, here there are individuals with particular symptoms. Doesn't feel quite right. This may be something new. So then what is the next step? How do you figure out that it's a virus, first of all, and then what kind of virus it is?
Yeah, well, the first thing they did was to try to grow the virus. So they had just a laboratory where they would routinely grow viruses, and they took some samples from the patients, basically put them in a petri dish, and saw that some of the cells started dying. And that was an indication that a virus was there.
Yeah.
Once you have suspicion for a virus, you can use one of my favorite techniques. It's called metagenomic sequencing.
Okay.
And it's a really good technique for finding out what might be there when you don't have any idea what it is.
Okay. Yeah, so metagenomic sequencing, that's a long word.
Yeah.
Two words, actually. What does that mean?
Yeah. Well, meta is kind of meta like everything. Genomic is obviously the genome of a pathogen or a virus. And sequencing is an approach that we use to take material from a sample and figure out what the genetic code is in that sample.
Yeah.
Usually when we do that, we know what we're looking for. So the human genome, we were doing sequencing of something we knew about, human genomes. When you do metagenomic sequencing, you don't know what you're looking for. So you actually take the sample, you sequence everything in it, and then you use sophisticated computer programs to figure out, oh, this piece of DNA or this piece of RNA looks like it might belong to a virus.
I see. And so that sample can be a mucus sample from a patient, but it can also be a sample of dirt in a forest. Yes. It can be a blood sample of a bat.
Absolutely.
So what are your samples? What do you look at?
Yeah, well, this brings us back to my earrings, Jaap. Ticks is one of the things that I'm most interested in. And actually, when you think about kind of the whole world of viruses, we know a fair amount about viruses that infect humans. We know a fair amount about viruses that infect other animals because we pay attention to livestock and things like that. But we really don't know a whole lot about what's out there in vectors.
Right.
And there was a paper a few years ago, one of my favorite papers, where they did a big sort of metagenomic exploration of invertebrates. So ticks, sea creatures, kind of any kind of animal species that you can imagine. And they found like 1,500 new viruses just using this approach. So I think looking at ticks, looking at mosquitoes, looking in these places we don't usually look can be a really great way to find out about new viruses.
So a lot of your work focuses on ticks. And so by looking at ticks, have you discovered any new viruses?
I'm sad to say that I have not yet discovered a new virus. It is a big hope and something that I feel pretty confident I can do in my career.
But isn't that kind of You want to discover something new and scary?
Yeah.
Is it not kind of comforting that you don't discover anything new?
That's a good point. The way that I think about sort of the fear factor here is that whatever is happening out there is happening, right? People are getting exposed to ticks. They're probably getting more infections than we know.
Okay.
And so finding something new isn't necessarily scary. It's just putting a name and a face to what might be happening.
Yeah.
But I do want to tell you about a virus that is of particular interest to me. It's transmitted by ticks. I didn't discover it, but I have taken a great interest in sort of raising public awareness of it. And that one's called Powassan virus.
Okay.
It was named after a town in Canada called Powassan. And it was actually found in the 1950s, unfortunately, in a boy who died from brain infection. And really not much was known about it for sort of decades later. And then it was actually kind of found again in the northeast of the United States.
Okay.
It was a different strain of the virus, and it was found to be transmitted by a really common tick, in fact, the same tick that transmits Lyme disease. So I think just like people are kind of getting exposed to Lyme disease and getting Lyme disease infection pretty commonly in certain parts of the country, I think people are getting exposed to this virus pretty often, and we don't know kind of the full spectrum of diseases that it causes. So I haven't sampled throughout the whole United States, but working with my collaborators in kind of the Northeast US, they've done extensive sampling in Massachusetts, in Maine, kind of other New England states, and even as far down as kind of New Jersey and Pennsylvania. And we do find the virus pretty consistently in ticks in those regions. I've looked a little bit here in Georgia, haven't found any so far. But there do indeed seem to be hotspots, which is areas where you can really find a lot of virus, and you can find the virus year after year. One of my projects right now is actually collecting blood samples from people who live in those regions, and we're looking at markers of infections.
And you presumably expect to find more than we know now.
Absolutely.
Yeah. Now, I know that in addition to being an assistant professor doing this research, you're also a medical doctor. So are you specialized specifically on infectious diseases or tick-borne diseases? And, through that, are you specifically aware of symptoms that could point you at tick-borne diseases?
Yeah, great question. So I do specialize in infectious diseases. Within infectious diseases, I have kind of a particular interest in brain infection because a lot of these tick-borne viruses can cause brain infection. I think this is the tip of the iceberg. I think that when people come in with really severe brain infections, we look really hard to find a cause of what's causing that. And so we're more likely to find something like Powassan virus. But if somebody comes in, maybe they have a fever, maybe they have a rash, they're going to be fine. Maybe we don't look so hard. And so that's one thing that I'm curious is Powassan virus could be causing that. I would say, though, kind of medical advice or just general life advice that I like to give folks is to really protect yourselves when you're out in the woods. Use bug spray, wear long pants and long sleeves so you don't get exposed to ticks. And then do go see a doctor if you develop any symptoms like a rash or a fever. I mentioned kind of this metagenomic sequencing is a really good scientific technique, but it's also started to be used more and more clinically. And so when someone does have nonspecific symptoms, there could be 100 things that it could be due to. Metagenomic sequencing is a tool that can help us find an answer more quickly than sending all of those different tests. Unfortunately, there's not yet a specific treatment for Powassan virus. There is a lot of research right now looking for antibody-based treatments. And you may be familiar with this from COVID because we used a lot of antibody-based treatments. And it seems like that may be a promising approach for things like Powassan virus. For the time being, prevention is better than a cure, which we don't really have. And so really, when we go hiking, be careful, don't get those ticks, and maybe leave them for you so you can discover new viruses. Exactly. We'd love to have them. Thanks, Jaap.
Okay. It was really wonderful having you on the show. We hope you enjoyed watching this episode, and we look forward to seeing you again.